Why don't we go ahead and get started? It's a pleasure to welcome our invited speakers this afternoon. And they are the dynamic duo of basic and translational research visiting with us from MD Anderson. And I was under the impression that they will give a duo in their talk, but the clinical talk was given, at 7 a.m., whatever, and this will be a basic translational talk. And this talk will be given by Dr. Keyomarsi. So I'm going to introduce both of them because they've worked together for about 25 years on breast cancer, drug resistance, and therapeutic strategies, and translational research, and it's really a great model for how that type of interaction and research can really have impact on the field. So Dr. Keyomarsi is currently a professor of experimental radiation oncology at MD Anderson, where she also holds the William Liedtke Chair in Cancer Research. Now, Dr. Keyomarsi has made her mark in part in the area of mentoring and training. And so she is the founder and director of the Triumph postdoctoral training program. She's co-director of the Catalyst Summer Training Program and director of the R25 Upwards Training Grant. And so she's very involved with training activities at MD Anderson. She earned her PhD at UCLA and did a postdoc with Art Pardee. How many of the students know who have heard of Art Pardee? Raise your hand. Okay. All right. Well, so Arthur Pardee described the restriction point of the cell cycle. He passed away a few years ago, and she was his postdoc, which is pretty amazing. And I was looking at her CV, and she had published about how you can synchronize cells using lovastatin, and published a little more on that later. But she's focused on cyclins, and most prominently cyclin E, isoforms, and amplification. And I did notice on her CV, she was on the original P21 discovery paper with Steve Elledge, CYP1. And so... I know. Well, CYP1 had to come from somewhere. So, has focused on various therapeutic strategies that have translated into clinical trials, not just with cyclin E, but with IL-6 and STAT inhibitors and autophagy in tumor survival, and WEE1, MYT1, dual inhibition, has a whole bunch of papers. And so I will say a little more about Dr. Hunt, just because they're both, because she's right here, and we may as well honor her as well. So she is the professor and chair of the Department of Breast Surgical Oncology at University of Texas MD Anderson, and a professor in experimental radiation oncology, MD from University of Tennessee, surgical residency at UCLA, and surgical oncology fellowship at MD Anderson. And her areas of interest include breast cancer as well as soft tissue sarcomas, with focus on targeting the cell cycle. She has over 700 peer-reviewed papers, 69 book chapters, 8 books. Importantly, she's immediate past president of the Society of Surgical Oncology and past president of the American Radium Society, which I had never heard of, but found it really interesting, and if you have time, go look it up. She's also 2024 Oncology Giants of Cancer Care. Congratulations, and a fellow of AAAS. So today we're going to hear from Dr. Keyomarsi on targeting the cell cycle machinery in cancer from bench to the clinic and back. But before we do that, I'm going to ask Ash to come up here and help me give them a little present.
Thank you very much. Thank you. Thank you. It's nice to be here. Thank you.
They're here.
Thank you.
Thank you.
The question is, can you post it before she starts talking?
Yes.
So thank you so much, Wafiq, for that very kind introduction. And let me do one thing. Okay, I just want to hide things. Okay, hide floating. Okay, very good. Kelly and I decided that in the past we've given our talks jointly, but since the work in our lab has really grown and I tend to speak faster, so I decided I'll give the talk today. So I'm going to talk to you about three different stories. The first one is targeting retinoblastoma protein in soft tissue sarcoma. The second one is combination of CDK4/6 and autophagy inhibitors in breast cancer that can actually induce senescence. And lastly, how targeting PKMYT1 in high-low molecular weight cyclin in triple-negative breast cancer can be efficacious. So the very first work was done by Dr. Tuyan Nguyen when she was a postdoc in my lab. She's currently a senior scientist at AbbVie Therapeutics in Chicago. So as you all know, let me just get rid of this thing too. Okay, as you all know, with soft tissue sarcoma, there's not that many options for treatment besides surgery, chemotherapy, and radiation. And tumors recur within 70 to 90% as locally inoperable metastatic disease. And the gold standard for treatment for advanced STS is different types of chemotherapy. And what's shown here is basically the same chemotherapy that's given in different orders. This is just the results of one of those clinical trials that compared either doxorubicin alone or doxorubicin with another chemotherapy. And you can see that the curves basically are superimposable, suggesting that there's really no benefit, additional benefit by adding more chemotherapy, with the median overall survival being 12.8 months. So we need novel therapeutic options. So we are kind of biased in our lab. We think of cancer as being a disease of the cell cycle, and the target for that, the cell cycle, is retinoblastoma, which is a tumor suppressor that regulates the cell cycle. So this is just a simple cartoon showing that how aberrations of our RB pathway occur in sarcoma, and this also occurs in other tumors, but I'm giving sarcoma as an example. So under conditions when RB acts as a tumor suppressor, it binds to E2F, and E2F basically sequesters RB. By the way, this line is a restriction point. However, when cyclin D CDK4/6 complex phosphorylated RB, E2F is released and then allows RB to come off in a hyperphosphorylated form. E2F then binds to the transcriptional part of a lot of the S phase genes and allows the cells to go through the cell cycle. What naturally inhibits CDK4 cyclin D is P16. However, what we see in soft tissue sarcoma is a deletion of P16. We can also see overexpression of CDK4/6, and we can also see overexpression of cyclin D. And under these conditions, when you don't have P16 and you have these kinases and the cyclin D being upregulated, they can push this pathway forward into S phase. So then come in the CDK4/6 inhibitors that are several of them. There are 3 in the class: palbociclib, abemaciclib, and ribociclib. They're cytostatic agents. They can actually inhibit this process, and by inhibiting it, it allows RB to now become a tumor suppressor again. And however, this activity is reversible. Part of the reason is that the CDK4/6 are in fact cytostatic agents. So we wanted to take advantage of this fact that if a cell has a retinoblastoma that has the ability to be phosphorylated, it can also be inhibited in a transient way. So our hypothesis is that what if we could reactivate RB as a tumor suppressor in cells and then sensitize those to DNA damaging agent? So the process has 3 steps. In the first step, we need to cause G1 arrest using a CDK4/6 inhibitor. Then we take that CDK4/6 inhibitor out, allow the cells to kind of synchronously go through the cell cycle. The cells, they're released from G1, and then they are then targeted at S phase with a chemotherapeutic agent. So we give CDK4/6 first, stop, allow the cells to go, and then by this time, they should have a much higher S phase population. We also need to have ways of monitoring this. So we came up with 2 different ways. One is PET imaging, the other one is circulating thymidine kinase. I'll go over that shortly. So we had 3 goals in our study. The first one is to identify RB as a stratifying biomarker for cell cycle targeted treatments, evaluate its therapeutic efficacy of sequential combination treatment, and then develop monitoring strategies to non-invasively predict this. So the very first thing we wanted to do was to say, okay, is RB even a player in sarcoma? So sarcoma comes in many, 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 many varieties. There are about 50 different subtypes. Here I'm just showing a few. And at MD Anderson, with collaboration with Alex Lazar, we were able to examine 1,000 of those patients for RB status. And what we found is that on average, 50% of the patients have RB being expressed, 50% don't. This shows a distribution in different subtypes. So the next question we ask is that, okay, we know what the RB status is. These patients, we also know what their clinical outcome is. Can we stratify these patients' outcome as a function of RB? And if we do, does RB make a difference? And the answer was no. So here it's showing 3 different subtypes, and it's showing which ones. So one of the red is RB positive, the blue is RB negative. And for the most part, the outcome data is superimposable. And the reason is that these patients did not get treated with an RB-specific agent. They got treated with chemotherapy. Chemotherapy cannot differentiate whether the cells are RB positive or RB negative. So since we could see this difference, we then wanted to see whether, in fact, if you take an RB positive cell and provide our treatment strategy to it, would they respond differently? So as far as the first aim of the study, we could say that RB is a substrate for CDK4/6. So then we hypothesized that RB positive soft tissue sarcoma would be good candidates for the CDK4/6 based therapies. So we did, the next thing, which is going back to cell lines. We had a whole bunch of cell lines that were RB positive, couple that were RB negative, and the rest of the cell cycle markers are shown here. We then treated them with abemaciclib, and we found that in all the cases that were RB positive, they responded pretty well with a low IC50, but if they're RB negative, they don't. And in fact, we could do a Pearson correlation with RB status and a response, and we could see that RB and the 2 different phosphor RBs, they're both pretty good markers for response to abemaciclib. So next we said, okay, if that's the case, what happens if we now take one of the cell lines and get rid of RB? So we did that with CRISPR-Cas9. We took here HC1080, it was RB positive. With CRISPR-Cas9 did, and then the very first question we asked, so whenever you do a CRISPR-Cas9 with any cell cycle genes, the very first experiment should be, does that affect the cell cycle? And it didn't, which was good. And we wanted to make sure that the cell proliferation didn't change. Again, we're targeting the cells with a cell cycle inhibitor. So we can't have a cell that has a lower cell cycle proliferation. So again, the cell cycle was great. There was no difference in it. However, these cells, when their RB was knocked out, they did not respond to abemaciclib. You can see that by the increase in IC50. We did the very first same experiment with P53 knockout, took the same cell lines, knocked out P53, and again, the cell cycle doesn't change, the proliferation doesn't change, and these cells did not, they all were responding to abemaciclib, suggesting that the action of abemaciclib is P53 independent, but RB dependent. So next we said, okay, we did the first part. We can cause the cells arrest in G1. What happens if you remove the drug? So we did time course experiments. Here we took, again, HT1080 cells, and we treated them for a couple days, and then we took time points every 3 hours. And what you can see that as the cells have been released from arrest, their S phase is their G1 first increases, then decreases, and S phase basically goes up. There's no change in G2. Here's just looking at the 2 time course, the 0 hour and 18 hours. And here, if you look at the orange S phase, you can see that there's a huge difference in that 18-hour period, suggesting that the cells are being very nicely synchronized. They're coming out. And here we can see that under these conditions, both phospho-RB and TK1 levels are also cell cycle regulated, the levels come back up. So this was the first indication that thymidine kinase could in fact be used as a monitoring agent. Here's looking at 2 different cell lines that did not have any RB. And the same strategy did not give rise to any changes in the cell cycle. In fact, if you recall in the previous slide, I went to 0.2 micromolar only. In this one, we had to go to 10 times higher, or in this case, 0.5 micromolar. There was absolutely no change. So now that we've done this, the next question is what happens if you now, you know when the cells go into cell cycle arrest, you know when the cells go into S phase, what happens now if you not give them the DNA damaging agent? So we use 2 different approaches, a sequential and concomitant. Sequential is showing abemaciclib first, then we then release them, then we give them gemcitabine, then we wash everything out, and then we do cell viability at the end. Concomitant is when you give the 2 together, and then you also then release at this point and the cells go out. So when we do this, we are able to examine combination index in the form of isobolograms. There are no other ways of doing it. And if our numbers were above the line, we would get antagonism. If it's below the line, we would get synergism. So in this case, drug A was always abemaciclib for 6 days. Drug B was gemcitabine. And we also did V1 kinase inhibitor. And what we found is that for the most part, when you gave the treatment sequentially, we always got synergism. When you gave the treatment concomitantly, we got antagonism. So we did this 100 different ways in 100 different concentration with different cells. And we did both sequential and, yeah, we did both sequential and concomitant. And because we had so much data, we basically decided to present it as a heat map. So synergism is always red. Antagonism is blue, everything between is additivity. So here you can see that if you take cells, HT1080 in this case are RB positive, or SKLMS are RB positive, and you treat sequentially, they're extremely synergistic. If you now take the other cells that are RB negative or RB low, none of them are synergistic. So this is sequential for all the cells. What happens now if you take the same cells that were synergistic here, and you would treat them now concomitantly, they're no longer synergistic, they're antagonistic. So this was, and we did this both with gemcitabine, and we did this with V1 kinase inhibitor. We got the same exact results. And here's just putting it together. You can see that this is what happens sequentially in blue, and in green is concomitant. Again, the only difference between what we're doing is not the dose, it's not how long, it's the timing, when we are giving drug A, when we're giving drug B. And this is taking advantage of the biology of the cell. What happens to these cells?
Can I interrupt?
Yeah.
So do you see kind of when you do a selection, you do for a sequential, is there a selection going on and you eliminate one population of cells and then another.
No, the cells are just, they're just synchronized. That's all it is. They're synchronous. You don't get rid of any cells when you first treat them with Abema and then let them go. It's the same number of cells.
So have you reversed the sequence?
Yes, we've done all of those. And there's no, it always has to be in this direction. And it has to be RB positive. We've done this in vivo as well. I'll show you the results, okay? So, so here is looking at what happens to the cells in the sequential setting. The cells undergo apoptosis. Concomitant, no. Single drug, no. And then so we've I've showed you the efficacy of the combination treatment in vitro. Now the next thing was to do it in vivo. So that was a little bit more difficult. For the in vivo, we gave abemaciclib 75 mg per kg PO every day, and then we recovered. We took the mice at different times, and then we examined proliferation, looking at BRDU. Later on, we actually did PET imaging, and we did circulating TK. So here's the animal experiment. So we give abemaciclib so there are 4 arms. A to G means Abema first, then you remove the drug, you let the tumors, the mice go for a couple of days, then you give them gemcitabine. Concomitant is at the same time, Abema alone or gemcitabine alone. So I'm going to ask you to look at this first. So this is the first cycle of treatment. And for the first cycle of treatment, this is looking at the tumor growth. And you can see that percent tumor growth. So the higher the number, the more tumor growth inhibition they're getting, I'm sorry. So here, gemcitabine, not that much tumor growth inhibition, abemaciclib, a little bit more tumor growth inhibition. This is what happens sequentially. Sorry, this happens when you give them concomitantly at the same time. You still don't see much, but when you give them concomitant, sequentially, you get this. So this is the orange one. So this is so this is just one cycle. We did this for 3 cycles. And the different lines are superimposable. So those are single agents alone or concomitant, no difference. The orange line is sequential. Again, the only difference is when we treated the mice. We did not treat them a different dose or anything. It's just different times. And this is what you get as far as survival. We see this in multiple models, including a PDX model from a patient from rhabdomyosarcoma who did not respond to chemotherapy. We see the same delay in tumor growth and also in the ability of tumor inhibition. And
are heavily
yeah.
tumors.
Yeah, again,
Very, very resistant tumors.
tumors, yes.
Did you reverse the sequential or the
Yeah.
gemcitabine and
There's no difference. That's in the paper, but we've done it in all directions. Yes. With these two, with these. Yeah, that's the point. The orange line is when you give them sequentially. No, we don't. This is continuous treatment. So this is multiple cycles. So we start treatment from here. This is, so we start, the reason the arrow is here is to show that at that point we could do statistical analysis because all the arms were active. Okay.
You may have said this before, but was there anything special about gemcitabine?
Yeah, at the time, because Lilly had both gemcitabine and Abema, and we wanted to work with them. But you can do this with any chemotherapy. We did the B1 kinase inhibitor. It works. Any drug, in my opinion, even though we only have used those 2, that is a chemotherapy. Any S-phase drug would work. Yeah.
And because we were using gemcitabine clinic for sarcoma
Yeah.
patients.
Yeah, that was the more important point.
Yeah, so we wanted to make sure that it was clinically sort of relevant to do a clinical trial after we had the proof of concept.
And here's also looking at gamma H2AX. You can see that in the sequential, you see a lot more brown compared to concomitant, compared to gemcitabine alone or abemaciclib. So, again, the combo treatment works in vivo. So next, how do we non-invasively examine it? Again, you can't take patients and get one of them and, do biopsy and do BRDU. So we had to have a non-invasive way of examining them. So we did two different methods. One is PET imaging with FLT. The T stands for thymidine. So you may all be more familiar with FDG. D stands for the glucose part. Or so we wanted to look at proliferation, and then circulating thymidine kinase is an assay that actually measures free thymidine kinase in the plasma. And what we did here is a PET imaging. I call this my $18,000 experiment. A lot of mice, this is in that were transplanted. We did their RB positive or RB negative tumors, and then we treated them. And then, so this is 6 days on with drug, then 3 days off and 6 days off. And if you then measure the amount of signal you see in the little circles, you can see that at 6 days, everything goes down. And then when you move it up, when you get rid of it, basically you can, the uptake goes up. This happens in the HC1080 with the abemaciclib. This is with vehicle, and this is with the knockout models. We can also see the same thing when we do it with plasma thymidine kinase. And this is really incredible because you just take the plasma from the mice. It's very non-invasive. And again, we can see that during the on days, it's gone. And we did two different concentrations of abema. And you can see that during the on days, it's gone as far as TK levels, but at 3 days off and 6 days off, they come back. And this is kind of looking in the graphic form. So we formulated the monitoring strategy. So then we took this and with Kelly, and then we talked to our sarcoma oncologist. And we went over kind of the strategy that we did, what the strategy was, that we did the in vitro, we did the in vivo, we did the monitoring. And we basically told them that that we've successfully completed this. This is a thought process that was at the lab, has nothing to do with new drugs, has to do with the biology of the cells. And we want to see whether this can be done in a clinical trial. And both Nita and Elise were very enthusiastic. They wrote the LOI, and they went to CTEP, and CTEP actually approved it, sponsored it. The clinical trial is now accruing patients, I think, from next week or beginning of March. So we're very excited about this. It took a year of just working with CTEP to get this done. And what the clinical trial is in multiple parts. So in the first part, all we're doing is that we're giving the patient abemaciclib, the same dose, and then we're doing PET imaging and TK at different points, either 8 days or 10 days. So for us to see at which point the FLT is going to come up or the TK is coming up. The phase two, the part B is when we then add the gemcitabine, and we have different doses, 3A, 3B. And we also take, and again, we do abemaciclib depending on what part A is going to show us, and then we add gemcitabine, and we're going to repeat the sequence until we get response. And of course, what CTEP wanted us to do is to have a control arm, and the control arm is basically gemcitabine and docetaxel. It's going to be arm A is our treatment part, and then arm B is the standard of care. And then we will cross over if one or the other one does not respond. So we have a goal in place. So that's kind of the end of the first part. And are there any questions before I go to the second? Yes, ma'am. Yeah,
Okay,
sorry.
What are the side effects? Is there any side effects in mice for the
Abemaciclib? No, not doing this. It's only 6 days.
Yeah.
So, and we're not giving the drugs at the same time. So we don't have any overlapping toxicity.
But, and then do you foresee any, like, has this been put in patients before? What is it? It seems like it would be pretty gentle, especially if it's RB specific, but.
Yeah, abemaciclib has been given to hundreds, probably millions of patients by now.
Yeah, it's mostly been used in breast cancer, FDA approved for advanced breast cancer. And it has been used in sarcoma. Sarcoma is more rare, and so it's not an FDA approved indication for the drug, but it is being used. And then gemcitabine, like we mentioned before, is standard for sarcoma.
Yes.
Does this have any applicability to pancreatic?
It can be applied to any cancer.
malignant side of the
It can be to triple negative, you know. My problem right now is to convince one of my postdocs to do something that's already been published in a different cancer. Yes,
Really interesting. So, you know, if you just wanted to try this approach in tissue culture in the lab
right.
as a new approach, I noticed you're treating your cells for 7 days to cycle.
Yeah,
Is that
six,
sort of
yeah.
standard?
Yeah. The standard is it's
Is
such
there
a
some-
static agent.
Is there something you're looking for to get to some endpoint to say, okay, now it's. the-
Yeah, it's G1 arrest. We basically, just G1 arrest.
File up in G1.
That's it. That's it. And then you have to take time points to see that it actually, it will reversibly go back up. You don't want anything to be in G1, you know, forever. So you want to make sure that, so what we do also is that we say, okay, what's the length of the, what's the doubling time of a cell? That's why I did all those doubling times. The doubling time is 36 hours. From the time that you basically release the cells to the next cell cycle should be the same number of hours. So you don't see any additional delay.
Well, I may follow up and chat with you.
No
Because
problem.
We are doing mostly DNA damage response modifier,
Mhm.
a lot of them work in S-phase.
This could actually work nicely. You'd be interested in my third VNet. Yes, sir.
So again, curiosity again, so you just said, you know, honestly, the cells in a particular cell cycle basically keep a sequence of treatment work.
Right.
And then, but how, you also said when you switch the sequence, instead of it followed by the target therapy.
Right.
It works.
It doesn't. No, it doesn't work if you reverse it. The only combination works is sequential abema first. That's the only combination that works. Okay. No, we've tried both ways at the same time. Yeah, it doesn't work the other way.
Release them, and then keep.
Yeah. So when we first proposed this, they said, no, they said, you cannot do this in vivo. So Lilly wouldn't even talk to us until we could show this in vivo. We showed it in vivo, then they came back and said, well, how the heck are you going to monitor it in patients? So then we monitored it in patients and then CTEP got interested. So, yeah. Yes.
Yeah, I don't know if I caught it correctly, but in one of your cell studies, it looked like there was a dramatic lag and delay for the sequential treatment in terms of inhibiting proliferation. But then it looked like it did start.
It does go up, yeah.
And so I wonder what percentage of those cells are escapees from the sequential regime?
No, it's sequential, they don't escape. It's the abemaciclib that's the one you wanted to escape. So our S phase goes from say 1% to about 20%. Yeah, they would following abemaciclib, of course, because you're removing it. They don't have any more selection. Yeah, okay. All right. Oh, sorry.
And I'm sorry if I missed this, but you said that the thing is also doing this in RB function.
Yes.
Only in cells, so in patients where it's not heterogeneous, that is.
Very good question. So I forgot to mention, so very good question. One of the first things we're going to do is to screen all patients for RB status. So that's the very first thing we're going to do. The beauty of it is that in the lab, you're working on a treatment strategy that's towards RB negative. So we will get the patients, we will know, we have this knowledge, all the patients who are RB positive, all the patients who are RB negative, so we can figure out what the difference between them are. Very good question. All right. So second story is a story that a graduate student in my lab started, Smurthy. And then her current position, she's a director and head of solid tumor lung oncology and discovery at J&J. Very proud of her. So she started this, so this was her thesis project. So now we're going to switch to ER positive breast cancer. So ER positive breast cancer, this is a history of how the treatment has evolved over the years. It initially started with doing mastectomies, and then we had different drugs that came in. We had tamoxifen, then we had the aromatase inhibitors, then we had fulvestrant come in, everolimus, and then in 2015, we had the FDA approval of the 3 CDK4/6 inhibitors, and now we're also giving PARP inhibitors and immunotherapy. So what has happened with the CDK4/6 inhibitors is that they actually have increased the overall survival of patients. Most of the patients actually now they have increased their overall their progression-free survival by 2 years, but you still have some patients that don't respond. So the three classes of CDK4 inhibitors are palbociclib from Pfizer, ribociclib from Novartis, and abemaciclib, I've been telling you, from Eli Lilly. All have been FDA approved in different conditions, but all of them with ER-positive disease. And the approval status basically started in 2015 and ended in 2018. This is standard of care right now for metastatic breast cancer, for ribo, and abema, it's now being used in the adjuvant setting as well because they also show overall survival benefit. So, so I'm going to concentrate mostly on palbo for this study. In phase 2 and 3, it prolonged PFS for more than 10 months, awarded FDA approval 2015. However, no reliable markers to predict response to palbo except for RB. And about 25 to 30% of patients do not respond initially. They have intrinsic resistance, and majority eventually develop. So develop resistance. So our goal was to improve the efficacy and selectivity of CDK4 inhibitors in breast cancer. So initially what we did is that we basically, took breast cancer cell lines and treated them with palbo for different days and then compared these to the normal cells. This is something I'd shown you before, and the cells basically arrest. And this is looking at what happens at a higher dose. Again, the tumor cells respond very nicely. So then we said, what happens if you treat them for 6 days and then remove the drug? Do the cells come back up? And the interesting thing was that, yes, that the tumor cells are coming back up when you remove the drug here. Again, this is just palbociclib. The question is, why are they coming back up? The MCF10A, all of them came back up. So one of the things we wanted to examine is that we wanted to see what are some of the biological effects palbociclib has on the cells. And we could see that at high doses, 5 micromolar and 10 micromolar, the cells actually underwent senescence. This is tumor cells. Nothing happened to the normal cells. And this is just another way to show senescence. So, however, at low concentrations, and this is kind of where biology comes in, and I never forget that day. So Smurthy was looking at the cells under the microscope. I told her, look at low doses. I want to see what the cells will do. So she was looking at the cells under the microscope and she was just taking way too long. So then Jason, who was another postdoc in the lab, she said, what the heck's, what's taking so long? She said, these cells look weird. So Jason looked at the cells under the microscope and looked at Smurthy and he said, I bet you anything, this is autophagy. So that was the thought process. And his thought process for autophagy is that the cells are getting bigger. So we thought it was a reasonable hypothesis to test. So we started looking at autophagic flux. And the way you look at autophagic flux is to look at LC3 and P62. And when you do a ratio of those, anything that goes up is suggesting that you have autophagic flux. And lo and behold, we had autophagic flux at low dose, not at high dose. And so then Smurthy did the next thing. She looked at EM. And when she looked at EM, you can see these double-sided membranes in both tumor cells that responded to palbo. So what is autophagy? Autophagy is there has several regulators and inhibitors, and it's a way for this, it's a survival mechanism for the cell. It basically takes the garbage that the cells make and gives it back to the media. So the cells provides amino acids to the cells, increases metabolism. So it's a one way for the cells to come out of it. However, there are inhibitors to it. They're both genetic inhibitors, Beclin and ATG, if you knock them out, and there are drugs that can inhibit it. So what we did is that we took MCF7s and T470, we knocked out Beclin and we knocked out ATG. So now we are getting rid of autophagy. Under these conditions, we asked, would palbociclib be more effective? And the answer was yes. So this is a simple dose response curve that shows what happens to the cells when they have wild type ATG and Beclin. And when you get rid of them, the IC50 shifts to the left. And then we did the same delay experiment that we basically removed the drug in these cells. And we could see that when you remove the drugs and they don't have ATG or Beclin, the cells don't come back up. And on top of that, these cells now at 1 micromolar can induce senescence. Well, before they had to go to 5 micromolar. So that was genetic. We then, of course, we're interested in drugs. So we did pharmacological. What's a pharmacological inhibitor of autophagy? Hydroxychloroquine and chloroquine. So here we basically took the cells, we did the same delay experiment. Here we added hydroxychloroquine on top of palbociclib, and you could see that the cells did not delay. And here you can see that the combination, while hydroxychloroquine by itself or palbo by itself is not sufficient to fully inhibit the growth of cells, when you do them together, they are. And here also with hydroxychloroquine, the cells undergo senescence at 1 micromolar. And this is just another way of showing senescence. So we started this in vivo to try to come up with a dose finding study. So very simply, we injected MCF7s into mice, treated them with different concentrations of palbociclib over a 7-day period. And then we just measured growth, and we could see that here basically that the growth of the tumors went down during those 7 days as expected. However, at very high dose, the mice just couldn't take it. It was just too much. So we were very interested in the lowest concentration of palbociclib that we could use to cause autophagy. So for that, we first looked at BRDU, and we could see that here at 25 mg per kg, BRDU has some statistically significant effect, but not that much. But at the highest dose in tumors, you can actually see senescence. So we also got a dose response both with BRDU and palbo, senescence. But then we started looking at autophagic flux, and we found that 25 mg for 7 days was sufficient for RB to go down, for LC3 to go up, and P62 to cause these cells to have an autophagic flux at 25. So we found our dose, 25. So then we also took those tumors and we did EM on them. And here again, you can see the double-sided membranes. That is a telltale sign of autophagy. And we did some RPPA on this to show what the changes are. So then we did the real experiment. So the real experiment was, not that the other were unreal, but this is kind of the final experiment. So we basically treated the mice with combination of abema and hydroxychloroquine for 21 days. And then we released for 21 days. We wanted to see whether the effect is stable. And the results are shown here. So vehicle is blue, so they continuously grow. The shaded area is when we did the treatment. The treatment was concomitant, by the way. So hydroxychloroquine has a little bit of growth inhibition, and then the tumors go up. Palbo is the red one. It has some growth inhibition, but goes up. When you do the combination, the treatment response stays. And here's just looking at the waterfall plot. This is at the end of 21 days. This is at the end of 42 days. Again, suggesting that the combination is quite effective. So we also did we did RPPA on these to see that the cell cycle goes down, senescence goes back up under conditions where we had added hydroxychloroquine to palbociclib. So we also wanted to do this with a different autophagy inhibitor. So at the time, and still, unfortunately, lyso-5 was a preclinical drug, is still a preclinical drug, but it's the next generation autophagy inhibitors. We got the same exact response. Again, this is the same experiment. I won't go over the arms. You can appreciate that the green arm shows a lot more response. And here's looking at waterfall plot and survival, that basically with this tumor that is very fast growing, we can get the tumors to respond. So what we show is that under conditions where, and the cancer, so what I didn't go into for time was that they have to be RB positive and cyclin E low. Under these conditions, when you treat the cells with CDK4, the cells on CDK4 inhibitors, the cells undergo autophagy, they survive. But when you add an autophagy inhibitor, they actually die. Kind of unfortunately what happened to the plane. So the preclinical work is that the conclusion from it is that palbo has a biphasic effect on ER-positive breast cancer. Low doses cause autophagy, high doses cause senescence. The inhibition of autophagy with both molecularly and pharmacologically sensitizes the cells. And palbo can synergize with autophagy inhibition in vivo, and there's synergism between the two. So we wanted to translate this to a clinical trial. So again, we went to our breast medical oncologist, Debu Tripathi, showed him all the data. He asked us to do some other controls, which we had done until we got convinced that this is good enough for it to go to a clinical trial. We wrote actually a grant, got funded by CPRIT, IRB approved. We did a phase 1 of hydroxychloroquine and palbociclib and letrozole, so triple combination before surgery in participants with ER-positive breast cancer. The study was finished. The paper just got published in NPJ earlier this year. And the PI, as I said, was Debu, and I was the co-PI. So our primary, so it's a single center, open label, non-randomized standard 3+3 dose escalation design. The primary objective was to evaluate the safety and tolerability of combining HCQ with continuous low-dose palbo and letrozole in patients with hormone receptor positive metastatic breast cancer to establish the recommended phase 2 dose. Secondary is to determine the response rate and clinical benefit of just 8 weeks of the assigned dose. So again, this is how the trial was done. We started with 400 mg per kg hydroxychloroquine. The dose of palbo was low. The dose in the clinic is 150. We used 75, but we did the continuous. Letrozole was the same dose. We increased, so as patients would not have any adverse events to the first dose of HCQ, we would go to the second dose. We then went to the third dose and expanded that. So then we had enough patients to cover each dose. And this is basically the how many patients we accrued and what the different exclusion criteria was for each one. This is kind of what the patient characteristics was for the most part. None of these patients had seen CDK4/6 therapy, but they had seen other types of therapy. So that was one of our exclusion criteria. We couldn't have any one of them had progress on CDK4/6. So this is just showing the adverse events. And the way we've done this is to look at the different drugs. So the first drug is palbo, is green. The second drug is hydroxychloroquine, is in blue. And sorry, the first drug, the other way, HCQ, I can't read my own. The first drug is HCQ, then letrozole and palbociclib. So here you can see that for the most part, just grade 1 and 2 toxicities or anything, palbociclib has the most toxicity on the patients. Then we also looked at just the unique adverse events just for hydroxychloroquine, and for the most part, and what each one of these colors shows the different doses. So you have so 400, and then grade 1, grade 2, grade 3, and then 600 the same, and 800. So again, most of the adverse events were grades 1 and 2, so very well tolerated. So we met the first objective. How did the patients do? Usually you don't do this in phase 1, but we actually got really nice outcome data. Only one patient progressed. All the other ones either had no change in their, so they had stable disease, or they had PR. Thank you, partial response. So 2 of the patients were doing so well, we continued giving them hydroxychloroquine for a year. And their tumor just basically stayed at bay. We also looked at biomarkers, so we can see that the patient 5 who progressed had no RB, and cyclin E was high, but in the other ones, you can basically see that the patients who had either partial response or stable disease had good amount of RB and also had low levels of cyclin A. We also looked at it. We had one patient that we had longitudinal biopsies for, and you can see that the autophagy markers in that patient go up on treatment as expected. And again, Ki-67 goes down on the same patient. So with that, oh yeah, and then once these patients came off treatment, these patients get heavily treated. So even after they went off our clinical trial, some of them went on to 7 additional clinical trials post our trial. So the conclusions we reached from our clinical trial, it was safe, it was efficacious. We came up with a recommended dose, which is 800 mg per kg. Activity, it inhibits autophagy, enhancing palbociclib efficacy, and we can reduce it that way. And we also identified biomarkers. So what we want to do is now we want to do a phase 2 validation study in a randomized trial to confirm efficacy, safety of biomarker correlations of HCQ, either palbociclib or the next generation CDK4 inhibitors with letrozole. We are looking at novel combinations, so next generation CDK4 with HCQ, and there are several drug companies that have that. So with that, I'm going to move to the third one, and if we could just wait for the questions till the end, that would be great, just because I want to. The last one is also very special, partly because one of my postdocs, Dr. Amirti Lula, got her PhD with Dr. Wafik El-Deiry. Her current position is a clinical scientist at MD Anderson, and the other person who led the work is Mili, who's a postdoctoral fellow. So what I'm going to talk about is the work that we've done for years and years and years, is looking at the low forms of cyclin A. One of the things that Kelly and I have done over the years is identify that low forms of cyclin are in fact a really good prognostic indicator for breast cancer. We looked at thousands of patients. This is the results of 3,000 or 4,000 patients looking at HR positive, HER2 positive, or TNBC. And you can see that whenever we detect the low forms of cyclin A, the patients don't do as well, no matter what the treatment is. So we need treatment strategies that can target the low forms of cyclin A. So basically, it's our biomarker being the low forms of cyclin A, and we want to do a treatment strategy that can help breast cancer patients. How are the low forms generated? So you have the full-length cyclin A, that's everybody knows about this. You've seen this in your textbooks. Is a cyclin A that binds to CDK2, is found in the nucleus, but it has certain domains at the level of protein. We call this I actually used to call them the avocado motifs, because those are the amino acids that makes them susceptible for cleavage by elastase group of proteases. When elastase comes and cleaves them, they cleave these low forms of cyclin A, so I'm going to call them LMW cyclin A. They still have the cyclin box, they still can bind to CDK2, but what they don't have is what we call the nuclear localization signal at the N terminus. So when the nuclear localization signal is gone, guess what happens to cyclin A? It can go back and forth to the cytoplasm or the nucleus, and this allows us to identify cyclin A in the cytoplasm. So some of the things that we have found over the years with low forms of cyclin A, and I won't go through this, is they bind with much higher affinity to CDK2. They're resistant to P21 and P27. They are less sensitive to degradation due to their soft cellular localization, because the proteasome that inhibits them is the FEW, the FBOX protein that inhibits them is in the nucleus. And they can actually bind to different substrates in the cytoplasm. So we also, through the work that Mili had done, we have found that the low forms of cyclin A are much more prone to DNA damage. They don't undergo repair, they undergo a lot more DNA damage. The homologous recombination is much higher in cells that have the low forms of cyclin A that undergo replicative stress-induced cell death. So then we said, okay, so we have full-length cells that are happy-go-lucky, but the low forms are the ones that actually are very high in tumors. They're prognostic, and there's no treatment for them. So we wanted to go back and develop an experimental model that we could examine the function of the low forms. So we took a normal cell, in this case 7676, we CRISPR-Cas9 the endogenous cyclin A, and then we gave it inducible forms of full length and the low forms of cyclin A. The very first thing we did was to do some gene expression arrays, and we were looking for pathways that we could inhibit. And what we found was that under conditions when you had low forms, this is green line, but not full length, which is the red line, an enzyme by the name of PKMYT1 was very highly expressed. And this is another way of showing it. So what does PKMYT1 do? You guys probably have heard of V1 kinase. PKMYT1 is the cousin of V1 kinase. They do the same thing, but on the other side of the coin. they cause phosphorylation of CDK1. So when CDK1 is phosphorylated on either tyrosine 15 or threonine 14, it kind of arrests the cells at the G2M. That allows the cells who are undergoing replicative damage to repair their DNA. So that's why inhibitors against V1 kinase is used clinically, and now inhibitors about PKMYT1 are used clinically. So the differences between V1 and MYT1 is that V1 phosphorylates CDK1 at tyrosine 15, the nucleus. PKMYT1 phosphorylated at threonine 14 at Golgi and ER. So we have antibodies against both threonine 15 and tyrosine 14, and we can examine that. So that's exactly what we did. We took the cells that I was telling you about, we induced them with the low forms of cyclin A. This is over time, cyclin is being expressed, PKMYT1 goes up at the same time, and T14 CDK1 also goes up. This is just looking at the densitometry of that. And we also did them at the same time. We did both full length and low form on the same gel. And here's looking at the full length inducible, low form inducible. The low form inducible causes PKMYT1 go up, and only the low form causes the tyrosine 14 go up. None of the other cell cycle modulators are changed. So we then said, okay, if PKMYT1 is, if the low forms are in the cytoplasm and PKMYT1 is the cytoplasm, do they bind? So we did mass spec on it, and we saw that while both full length and the low forms can bind to CDK2, the low forms are the only ones that can actually bind to PKMYT1. So there's a reason for PKMYT1 levels to go up. We showed this with the IP Western in 293 T cells, and then we showed them again in our inducible model, and we showed them again in our immunofluorescence, where we can see both cyclin A and PKMYT1 are co-localized in the cytoplasm. And here's another way of showing full-length cyclin A, that you can see full-length cyclin A only in the nucleus. No PKMYT1 is being expressed. And here's the low forms of cyclin A that are in the cytoplasm, and you can basically see cytoplasm, and you can see PKMYT1 core expressed in the cytoplasm. And we can see that only the low forms, but not the full-length PKMYT1 stabilizes, only low forms, but not full-length cyclin A can stabilize PKMYT1. So the reason cyclin A binds to PKMYT1 in the cytoplasm is to stabilize it. And we could show this with cyclohexamide chase assay. So how about a PKMYT1 inhibitor? Well, there is one. It's called RP6306. The company Repair has identified it, and it's currently in different phase clinical studies, both as monotherapy in combination with chemotherapy, and in combination with V1 kinase, and in combination with ATR inhibitor. So the very first question we did is that if you take the low forms of cyclin A and now treat these cells with PKMYT1, are they affected? The answer is yes. And when they basically, in the absence of PKMYT1 inhibitor, as I showed you before, the phosphorylation of CDK1 goes up. But in the presence of RP6306, the levels of PKMYT1 don't change, but the phosphorylation of CDK1 does. And then we said, how about other triple negative breast cancer cell lines? So we have here some with the low form, some without the low form. We did similar experiment to what I showed you with abemaciclib in sarcoma, but in this case, we looked at PKMYT1 in triple negative breast cancer. And again, if they don't have the low forms, they don't respond as well, but if they have low forms, they do. And when we correlate this to either full length or the low forms, we can see that actually has very good correlation to the low forms of cyclin A and not full length. What happens to the cells when you treat them with PKMYT1? If they have high levels of cyclin A, they actually undergo apoptosis. They undergo G2M, and their chromosomes just burst up. So this is what happens to a cell that doesn't respond to RP6306. Normal chromosome, this is what happens to a cell that responds to RP6306. The chromosomes disintegrate. And this is looking at the number of breaks per cell. So then we did all sorts of experiments where we overexpressed the low forms in 231. They now are more responsive. We took away the low forms from MDA157. They're no longer responsive. We examined their gamma H2AX only when they had the low forms, do they respond? If you get rid of the low forms, they won't have high levels. Then we took a cell line that had amplification of cyclin E, and when we shRNA cyclin E, we still could see the full length, but not the low form. So this was a perfect experiment. And here we can see that under these conditions, these tumors that don't have the low forms, they don't respond. And we did DNA fiber assay to look at their replication fork trajectory. And we could see under conditions where they have the low forms of cyclin E. So this is this cell line. That the DNA is getting, the DNA fiber is getting shorter and shorter, but when you get rid of cyclin E, there's no effect whether or not you add RP6306. So then we said, okay, what happens if we put both the full length and the low forms in 231? And when you put the full length in 231, they don't respond, but when you put the low forms, they do respond. Again, it's low form specific, has nothing to do with full length. And the reason that happens is because when you have the low forms, the cells go into mitosis a lot faster. And this is looking at the ability or the timing of how treatment with with cells that have either the low forms or the full length go into mitosis. And this is as a function of time, again, much faster going into mitosis than the full length or the vector alone. So I will end with the work that we did with in vivo. The very first experiment we did was with MDA231s by themselves, no full length, no low forms. We injected them into mice, treated them for 40 days with 2 different concentrations of RP6306, no change. When you give them full length, they respond a little bit, but not that much. This treatment isn't that great. But when you give them the low forms, the treatment is black and white. And you can see that at the lower dose. We then did it in PDX tumors. We had a PDX tumor. This is from TNBC, heavily treated, cyclin E amplified. They respond beautifully to RP6306 as a single agent. This is another PDX model that did not have the low forms, and they did not respond. So, and then under these conditions, we ask what happens to the actual substrate, the substrate being the phospho-CDK1. Interestingly enough, in both high cyclin E and low cyclin E, phospho-CDK1 levels do go down, but it's only in the high cyclin E that basically the tumors respond and they undergo high gamma H2AX, and Ki-67 only goes down in the BCX70 model. Lastly, we did this in our transgenic mouse model. We've generated a cyclin E high transgenic model. And under these conditions, we can see that with the high-low form cyclin E, they respond. The phospho-CDK1 goes up. Phospho-CDK1 goes down. Gamma H3X goes up. And when we do this for a longer period of time, we can actually see that we have a very nice increase in survival and of the mice. So last but not least, how do we translate this clinically? Right now, we just have biomarkers. So we had patients that, we had a clinical trial with Kelly that it was neoadjuvant clinical trial that patients got treated with a neoadjuvant chemotherapy, and then we had taken samples from them. We had cyclin E levels on them already. So I asked what happens when we also look at phospho-CDK1 in these. And we can see that at least in this one tumor, that whenever cyclin E is high, phospho-CDK1 is high. And when we combined all our patients that we had available, we found that there's a very nice correlation with all the patients that had high low forms, this is in red, and high CDK1. So you can see that most of the patients that high low forms also had high CDK1. How about a response? We know that patients that have high levels of low forms do not respond to chemotherapy. And this is kind of what we saw, the pathological response. What happens to phospho-CDK1 in those settings is the same thing. Patients that have high phospho-CDK1 do not respond to chemotherapy. And this is looking at basically the 2 of them. So in this 19 patients of this group, both of them have both high cyclin E and high phospho-CDK1, suggesting that we can use these 2 as biomarkers, at least in TNBC, to treat them with PKMYT1. So the conclusions of this part, I showed you what the function of the low forms on PKMYT1 is, the effect of the cells in vitro and in vivo, and the fact that this translational significance is we actually identify them as biomarkers. And last but not least, this is our team. You need a new picture, but this is our team from a couple of years ago. I talked to you about the work that a lot of these people have done. These are the ones in blue, sorry, in red. The blue ones I hadn't had a chance to talk to you about. And again, we wouldn't be anywhere if we didn't do collaboration. We have collaborators in the sarcoma, in the breast, and of course, we wouldn't be able to do anything without biostatistical analysis. And on that, I'll stop and see if there's any questions.
Ash.
Regards to the last part that you mentioned, the low molecular weight cycling, given some of the functional redundancies between PKMYT1 and V1, have you considered using a V1 inhibitor in conjunction with a checkpoint?
Very good question. The answer is yes, and they're synergistic. It's gorgeous, yeah. Sure.
First is here, looking at segment E1 or also segment E2, and that's the difference.
Yeah. The easy answer is I don't know. There is redundancy between them, but we haven't seen cyclin E2 have the same prognostic significance, but we haven't looked at it that significantly just because we get really nice results with cyclin E1. So, but yeah, it's a good question. We haven't done it.
Secondly, your last project, you showed RB loss in conjunction with segment E2 with the functional relationship there. Or segment E just
Yeah, so it's not even RB loss. When cyclin E, the low forms go up, they bind a lot tighter to CDK2. That binding causes higher activity of that complex with CDK2. That causes RB to be phosphorylated. The phosphorylation of RB basically inhibits it as a tumor suppressor. Over time, tumors that have high levels of cyclin E could lose RB without any defect to the cells. Yes.
So, in your second talk, my understanding, the net outcome is cellular senescent subject.
Yes.
What is basically those cells that are come out, such as like, budding from molecular giant cancer cells to, for recurrence, or what is the overall fate of those senescent cells?
Dormancy, I would assume. We haven't done those. Those are very good experiments to do. We would require you to actually do the experiment, leave the tumors in there, and then see what happens. We haven't done that experiment, so but it's very, very good point. Yeah, I think dormancy is a definite outcome for some of the tumors that escape. Yeah.
You are considering using senolytic compounds?
We actually have done it with STAT3 inhibitor, which is a senolytic compound, another talk. So yeah.
Can you comment on immune effects with the CDK4/6 inhibitors? And can you tell our audience?
Oh, you're asking him or me? Oh, sorry, you're asking.
You.
I thought you were asking. No, just kidding. Just kidding. No,
And
no.
Can you give our audience some words of wisdom from your point of view of a mentor and a trainer in 2025 for our trainees?
So the second question is easier to answer than the first question. So the second question, as far as the trainees, each trainee has a different set of priorities and goals that may be very different than mine. When I first started my lab, I used to suffer from why can't everybody think like me syndrome, or why can't everybody work as hard as me syndrome? And it doesn't mean that the people in the lab aren't smart, or it doesn't mean that the people in the lab don't work hard. They just have different priorities. And sometimes they don't even know what those priorities are. And the important thing is to get into their heads and figure out what it is they want to do. And then make sure the work is interesting enough. And when they ask that they want to be able to do a certain type of work, and you say yes during the interview, make sure that you stay true to that promise, because sometimes, again, things in the lab could go in gazillion different directions. And I've done that myself. When I say something to the to the mentor, and then we never go there just because at the time there were other things that we want to do. So my thing is that if the student or the postdoc is here, they want to be here. They can do, you guys are all so bright, you can do anything you want. You can go conquer the world. You're here for a reason. So it's important for us to have that discussion. Now, to answer your first question, which is has to do with the immune function, absolutely. We actually think that CDK4/6 inhibitors do have an effect on the immune population, on the T cells, and our paper just came out in we did single cell sequencing on palbociclib resistant patients, and we found that, and what we're trying to do, we're trying to separate what it is in the immune microenvironment or the tumors themselves that would make the cells a lot more responsive to, say, CDK4/6 inhibitors. And what we found, if you take the cells at baseline, they have very good T cell response. So those cells do respond to palbo, but they could also respond to the combination, but they have to be at baseline. Those same tumors, once they progress, they lose that ability. They will have all these exhaustive molecules that they no longer are responsive to checkpoint inhibitors. So the question is that what combination of factors can you identify at the very beginning in baseline patients before you know that they're going to respond to palbo or not, to then combine them with both palbo and checkpoint inhibitors? Does that answer your question? Okay.
Yeah, sure. Thank you. Any other questions? All right. Thank you very much.
Thank you.
I have to go get my orientation.